All right, this video is on cellular respiration, which is chapter 2, section, or chapter 6, section 2, and chapter 6, section 3. We're going to go all the way in this video until we get to fermentation, which is going to be another video. Okay, so start us off. Let's go and take a look at what cellular respiration is. Okay, a lot of energy is released when the reduced molecules with a lot of carbon-carbon bonds, a lot of carbon-hydrogen bonds are fully oxidized, and the eventual result being carbon dioxide. Okay, so CO2. Okay, the oxidation occurs in a series of small steps. Those three pathways. The first one being glycolysis. And the second one being pyruvate oxidation. And the third being the citric acid cycle, or the Krebs cycle. Okay. We are going to go into more detail with the pyruvate oxidation and glycolysis and Krebs cycle in this video. So what we have right here is a graph showing how energy metabolism really works in our bodies versus, say, in a test tube. Okay. A is representing glucose and oxygen. If you notice, the free energy amount is on the same level for both A and B, but with A, there's small energy activation energies for each little reaction, and each reaction has intermediates that will eventually lead to the production of the product carbon dioxide and water. And if you look at B, starts with the same reactants, ends with the same products, but the larger activation energy is going to require a lot more heat. And this is why our bodies don't partake in option B, but more of option A, is because the small activation energies are going to be easy for our bodies to handle versus the high amount of activation energy that's going to need to go in for B. Okay, the amount of energy that's being reduced is going to be the same. Okay, but what is important to note is that the combustion, with an O, of a hydrocarbon is going to require a high activation energy, and that activation energy is usually going to be in the form of kinetic energy of heat. And those molecules move really fast. In order to get our reaction, or in order to get this reaction to take place in our body, we would have to have our body temperature reach highly unsafe levels to the point where we would die. Okay, but with these smaller activation energies, this little guy right here and this little guy right here, we can kind of take them one step at a time, and all these little intermediates that go in between, every single one, we're getting a little more energy, a little more energy, a little more energy, until we both end up with the same product. So this is catabolism versus the combustion of glucose. Okay, so this is a continuation, or not a continuation, but a summation of what cellular respiration is. Okay, we have actually 4 different stages. Okay, here's 1, 2, 3, 4. Okay, and it's important to note what goes in. Okay, glucose goes in, and then carbon dioxide and water come out. And of course, there's going to be oxygen is going to need to come in for this. Just happens to not be during this stage. I'll go ahead and fix that. And it's not going to be during that stage. It'll actually be during this stage right here. This is where we're going to have the oxygen really have a role in the process. Okay, and then we're also going to have a lot of ATP being produced. Okay, this of course is, like I said, a quick summation. But the important part to know right here is that there are 4 steps, and this is where they all take place. Okay, so glycolysis happens inside the cytoplasm, or the cytosol, right? And of course, because it's in the cytosol and the cytoplasm, it's important to note that all organisms have the ability to undergo glycolysis, and they all do. The pyruvate oxidation and the citric acid cycle both happen inside the mitochondrial matrix, which is inside the inner membrane, because mitochondria actually have 2 membranes, an inner membrane and an outer membrane. And inside the inner membrane is the matrix. And that's where the citric acid cycle and pyruvate oxidation occur. And then on the inner membrane itself is where the electron transport chain is going to take place. And like I said, we're going to go into all bunch of detail for each of these 4 steps. Okay, so glycolysis, like we just said, takes place in the cytosol or the cytoplasm. Now, the final products of the glycolysis is going to be 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of high energy NADH. Okay, so what do we have here? These are electron carriers. These electron carriers are high energy electron carriers. Important thing to note about the ATP, with glycolysis, it actually took 2 ATP to start the process, okay, but it's going to yield 4 ATP. So this process makes 4 ATP, but it took 2 to start it. So that's why we say that there is going to be 2 final products of ATP. Okay, and then the pyruvate, which is going to be go in one of 2 directions depending on if oxygen is present. If oxygen is present, it's going to go into the oxidation process to get ready for the Krebs cycle. If it's not in the presence of oxygen, then it's going to start fermentation, which is another video. Okay. So, assuming that there is oxygen present, we are going to start the oxidation of pyruvate. In this case, there's going to be some products. We're going to have 2 products. We're going to have carbon dioxide, okay, is going to be produced, and this is going to be 1 carbon dioxide, 1 NADH, okay, but this is per pyruvate. Well, if we look from our last slide, that means, well, there's 2 pyruvates in every glucose molecule broken down. So this is actually going to produce 2 carbon dioxides overall and 2 NADHs overall while this is occurring. Okay, so the products, the CO2 and the acetate, is then bound to an enzyme called coenzyme A. Okay, so that's what goes right there. Okay, so coenzyme A is going to react with the pyruvate, and in the process, NAD plus is going to be reduced to form NADH, and then carbon dioxide is going to be released. This acetyl-CoA is going to go into the Krebs cycle, okay, which is going to be what we talk about next. So the citric acid cycle or Krebs cycle, okay, it's going to operate like we said before. The Krebs cycle is going to do a cycle for every 1 pyruvate that is made, which means, or in this case, because it's no longer pyruvate, now it's acetyl-CoA. For every 1 acetyl-CoA, it's going to go through a cycle. And since 2 acetyl-CoAs are made for every glucose molecule, this is going to happen twice for every glucose molecule. So we're going to have 2 circles. There's 1, and then we're going to do it again. Okay. Now, some important compounds. We know that acetyl-CoA, which comes from the oxidation of the pyruvate, and then there's also going to be oxaloacetate, which is going to be in the cycle, and it's going to react with the acetyl-CoA and start the Krebs cycle. Okay, I'm not going to need you to know all these compounds. Okay, but there's a couple, like I said, I think you should be aware of. One of them is the acetyl-CoA, and the other one is the oxaloacetate. Okay, because those 2 things are going to react right here, and that's going to be our starting point for the cycle. Okay, so as it goes through, eventually oxaloacetate will be reformed, and then it will interact with another acetyl-CoA and just keep going. So let's take a look. Remember, this is 1 acetyl-CoA is from 1 pyruvate, and there are 2 pyruvates that are made for every glucose molecule, which means there's 2 acetyl-CoA. So this is going to go through twice per glucose molecule. So keep that in mind when you are counting up how many NADHs and how many CO2s and how many ATPs are being formed, because you are going to be responsible for that information. Okay, so as we go through the cycle, okay, we'll go and go right here. All right, hey, first stop. Okay, we have the reduction of NAD plus to form NADH. Okay, again, since we're going to talk about, I'm going to talk about the whole thing as the whole cycle as per glucose molecule. So I'm really going to have 2 NADHs formed right here. And right here, I'm going to have 2 CO2 is being formed. Okay, I keep going. Right here, another redox reaction, and the NADH is going to be reduced, or the NAD is going to be reduced to form NADH. So I'm going to have another 2 NADH molecules here. So right now we have a grand total of 4. Okay, and then again some more carbon dioxide. Again, 2 of them. So it'll be 2 CO2 molecules. Keep going. Now another little step right here. GTP looks looks familiar. It looks like ATP, and in fact, it will convert into ATP. All right, but remember, you don't need to remember that, oh, the GDP is going to be converted. Just know that ATP is going to be produced in the Krebs cycle. And like we said, this is per cycle, so there's 2 cycles, so there's 2 ATPs being formed. Okay, continuing on, another new character. This is another high energy electron carrier. This is FADH2, and 2 FADH2 molecules are going to be formed. Okay. Back over here, we're going to have another redox reaction in which 2 more, this is, remember, per 2 cycles, because this is 1 per cycle, 2 more NADHs are going to be formed. So far, if you look, in just the Krebs cycle going through twice, we're going to end up with 2, 4, 6 NADHs, 2 FADH2s, 4 carbon dioxides, and 2 ATPs. Okay, so a lot of stuff's happening in the cycle. Make sure you keep track of your numbers. Okay, now the NADHs here and the FADH2s, those are all going to go to the electron transport chain. Okay, all the carbon dioxides, you know, they're going to be exhaled, and all the ATPs that are being formed, well, they're just going to be used for what we use ATP for, which is an energy currency. Okay, so like I said, important to note the introduction of a new energy carrier, and the NADHs and the FADH2s are going to go to the electron transport chain, which is our next stop. All right, so the electron transport chain, the NADHs and the FADH2s are going to be oxidized again to form NAD and FAD, and oxygen is going to be reduced to form water in a series of steps. There are 2 different steps here. Okay, one of them is going to involve the use of these electron carriers, so that's going to be the respiratory pathway, or the respiratory chain, and then we'll have the electron transport chain, which is going to be electron transport chain, which is the passing of the electrons from one protein to another inside of the membrane. Okay? Okay, sorry, just got this confused. Okay? These are the right words to put in here, but I just described the opposite one. Okay? The respiratory chains are involving the proteins and the electrons being transferred throughout the proteins, and then the electron transport chain is the oxidation of those high energy electron carriers. All right, so here's this heavy picture. Let's take a look. This right here is the inner membrane of the mitochondria. And here we have our high energy electron carriers. What's happening to them? They are becoming oxidized, which means these proteins right here, okay, these guys right here, they are being reduced. That's why they're gaining these electrons. And I'm going to go ahead and make these electrons red so we can follow them. Okay? All right? So those electrons that are being transferred into the proteins is providing energy to push these protons, these H pluses, across and against the concentration gradient. Okay, and of course this having a whole bunch of H pluses is going to end up making a high concentration versus a low concentration, which is going to be important right over here with this guy, the ATP synthase. Okay, so the FADH2 is going to do the same thing. It's going to transfer its electrons into the proteins, and the proteins are going to then use the high energy electrons to push more protons across. Okay, oops, that should be green. More protons across, and then finally, at the end of the line, at the end of this chain of proteins, we're going to have the reduction of oxygen into water. So the final electron acceptor is oxygen. Okay, that's where, talking about aerobic respiration, you need oxygen, there needs to be oxygen. This is where we see it. This is where it comes into it, because now oxygen is the final electron acceptor. Now, how does this all play? Well, as we built this high concentration of hydrogen ions on one side, or the outside of the inner membrane, okay, and then we have a low concentration over here inside the mitochondrial matrix, there's now a concentration gradient. So these protons are going to want to push back across. Well, in doing so, like we mentioned in a previous video, this ADP is going to be here, this P is going to be there, and the process of those protons coming across is going to cause a conformational shift inside the ATP synthase, and it's going to push these two guys together to make some money, make our ATP. Okay? And then that's how a majority of our ATP is being made. So the oxidation reactions, like we said earlier, are exergonic, meaning energy is going to be released during this process. Okay, so the energy is used to actively transport those protons back outside the mitochondrial matrix, and of course that's going to be setting up the potential for chemoosmosis and to send those protons back across. Okay, and so like I said, ATP synthase in the membrane uses the gradient of H+ to produce and synthesize ATP by the chemoosmosis. Okay, so important, about 32 ATP molecules are produced during the full oxidation of glucose. That is, we're talking about inside the electron transport chain. Okay, so 32 in electron transport chain. We're going to have 2 in Krebs cycle, 2 in the glycolysis process. So you're going to have a grand total of 36 ATPs being produced. Okay, so the role of O2, it's going to be that final electron acceptor. And what does that mean? It means that because most of the ATP is produced and formed by the oxidative phosphorylation, okay, like I said, and that's due to the oxidation or reoxidation of not just NADH, but also FADH2, FADH2 is going to be reduced. This is going to go into NAD+, and this is going to be FAD. So NADH is usually the more, it's a higher quantity. Okay, we're making more NADH than we are FADH2, but FADH2 is still producing and still providing high energy electrons to those proteins. Okay. All right, this is the last slide, and on it, it's going to be just a blank. Okay, I have the 4 stages. All right. Now, what I want you to do is for every stage, okay, so this isn't really a stage. Here's 1, 2, 3, 4. I want you to have some sort of flow chart or some sort of organizational picture or diagram, and it could be something as simple as just drawing arrows. I'm fine with that. But I want to see what's going into the process and what's coming out of the process. What's going in, what's going out. What's going in, what's going out. And I don't want to just know what, I want to know how much as well, okay, because that's going to be important. What goes in, what goes out, and hopefully we realize not just what goes in and what goes out, but also what's being recycled. Okay? So that's cellular respiration. If you guys have any questions, please ask.